Hi, today we will talk about recombinant DNA. Recombinant DNA is a form of DNA constructed in a laboratory. It is generated by transferring selected pieces of DNA from one organism to another. This process is known as DNA cloning, with the resulting cloned DNA often referred to as a recombinant DNA. Steps of making rDNA. DNA isolation. DNA is isolated in its pure form, which means they are devoid of other macromolecules. Cutting of DNA. For this step, the restriction enzymes are quite viral. It helps to identify the location where a designated gene is introduced into vector genome. Amplifying of DNA. Copies of gene are amplified through PCR or polymerase chain reaction. It is essentially a process of increasing a single DNA copy into several copies. After the desired gene of interest is cut with the restriction enzymes. Joining DNA. The vector and a section of DNA is joined in this step. It is achieved with the help of enzyme DNA ligase. Insertion of rDNA into a host. Here rDNA is added to the recipient host cell, and the entire process is called the transformation. Uses and aims of recombinant DNA. It also used to produce clotting factors of treating hemophilia and in the development of gene therapy. Recombinant DNA technology has proven important to the productions of vaccines and protein therapies, such as human insulin, interferon, and human growth hormone.